Hello, my name is Patrick Blevins, and today I'll be presenting on cross-presentation of exogenous antigens. This will be for exam 2B. First, we're going to talk about the introduction or the history of cross-presentation. Cross-presentation refers to the display of MHC class 1 peptides from extracellular acquired antigens. The process of cross-presentation was first recognized in 1976, though this process is thought to be a major means by which that dendritic cells can activate naive cytotoxic cells. The underlying mechanisms are still not well understood. Um, Cross presentation was discovered when researchers found that the CD8 plus CTL responses could be mounted to certain antigens that were known to be extracellular, and peptides from viral antigens could be presented on MHC class 1 even when the endogenous processing pathway was blocked. The phenomenon was called cross presentation because the viral antigen appeared to physically cross over from an infected host cell to an uninfected APC that presented peptides from the antigen of the MH class 1 as if the antigen had originated in the interior of the APC. Though there's been various APCs appear to be capable of cross presentation in vitro, the DCs or dendritic cells are the main cell type responsible for antigen cross presentation in vivo. Uh, Dendritic cell subsets vary in their ability to carry out cross presentation. Sorry, with resident dendritic cells located in the lungs and blood vessel walls being probably the most proficient. In contrast, though, the migratory dendritic cells are less able to cross present and concentrate on displaying peptides on the MHC class 2. There have been recent studies that show that DCs or dendritic cells are the most efficient of all the cross presenters. The next thing we're going to talk about are the mechanisms and functions of cross presentations. There are two possible models that have been proposed for how dendritic cells accomplish cross presentation. The first one hypothesizes that cross presenting cells possess special antigen processing machinery that allows loading of exo- exogenous sorry exogenously derived peptides onto class 1 MHC molecules. The second postulates that specialized endocytosis machinery that can send internalized antigen directly to an organelle where peptides from those antigens are then loaded onto one class 1 MHC molecules using conventional machinery. The means by which antigens achieve crossover from its exogenous or extracellular origins to the endogenous pathway are still unresolved. There are many instances where cross-presented antigens have been found to enter the cytoplasm. Uh, the book actually states that in the early 1990s, it was shown that the bead conjugated antigens captured by cross-presenting dendritic cells could reach the cytosol of these cells. Later, ML Lin and colleagues showed that exogenously added cytochrome C, which causes programmed cell death when present in the cytosol, could induce cross-presenting DCs to undergo apoptosis. It is now proposed that retrotranslocation or movement of the endocytosed proteins out of endocytic compartments and into cytosol can occur via TAP molecules present in these endocytic membranes. It's a sort of a black flow out of endocytic vesicles. However, TAP independent cross presentation of some antigens has also been observed, suggesting multiple mechanisms may exist for targeting externally derived peptides to class 1 molecules. So I got that example from the book. I thought that fit well right there. The next question we're going to answer is how can exogenous peptides be loaded onto MHC class 1 molecules? Sorry about that. APCs are antigen presenting cells most often acquire viral antigens by internalizing debris from the infected cells that have undergone necrotic or apoptotic death. In rare cases, the dendritic cells may also require portions of the membranes of live infected cells in a process known as nibbling. In any of these situations, because a viral protein has entered the antigen presenting cell or the APC from extracellular environment, it is initially directed to the endocytic system in the usual way. The viral peptides appear on the APC surface associated with MHC class 2, and a Th helper cell response to the antigen can be mounted. Uh, though during this initial processing of the viral proteins in the early endosomes, a fraction of the resulting polypeptides may be actively transported from the endosomes into the cytosol by endosomal membrane proteins, whose role in this translocation process is not yet clear. Um, in the cytosol, the polypeptides are taken up by proteasomes and degraded to peptides. Excuse me. The viral peptides are then transported via TAP into the ER or endoplasmic reticulum and loaded onto MHC class 1 just as if the viral protein had originated within the APC itself. This pathway has been dubbed the cytosolic route of cross presentation. Alternatively, there is some evidence for cross presentation by an avicular route that does not involve proteasomes. According to this model, some of the MHC class 1 molecules may be diverted from the endoplasmic reticulum into an earlier endosome where an extracellular acquired viral antigen is undergoing degradation by lysosomal enzymes. The combination of these diverted MHC class 1 molecules with the viral peptides within the endosome creates PMHC complexes that subsequently can be transported to the cell surface for antigen presentation. Lastly, a very few extracellular proteins appear to be able to cross the plasma membrane directly. They bypass the endocytic system entirely and enter a proteasome. Peptides generated via this latter mechanism could presumably associate with MHC class 1 via the cytosolic route. Regardless of the pathway of cross presentation, the end result is a display of viral peptide MHC class 1 complexes on the APC surface that can be inspected by antiviral CD8 plus T cells. Antigens from intracellular bacteria and parasites can also be processed, and their peptides displayed on MHC class 1 via cross presentations. Um, these are the references that I used uh, for this presentation. And Dr. Blair, I'm fine with you using this video however you need to. Thank you.